Okay, brilliant. So we are coming close to the end. We have now identified a new gene that may have something to do with ATR and may point to a new biological, connection. So that's really exciting. However, this is unexpected and this could be still some random noise. So even though we've been quite stringent, you have seen the Spearman disagreed with the Pearson. We're not sure, but so what do we do next? So one thing we can do, and this is what we are going to do, is we're going to look at the networks of both ATR and SLU7 and see whether there's an overlap. And these overlapping genes then will point us whether there's really a pathway connection between the two genes. And that will be the final task in this particular project. So just to explain you this rationale a little bit more in detail. So we have a set of genes for ATR, and SLU7 is one of those that are correlating with ATR. So this is a group of new genes that, and also known genes that have some connection with ATR in terms of their gene dependency correlations. But now we can do the same analysis again and collect the genes that are significantly correlated with SLU7. And now if only SLU7 is in the intersect itself, so if there's no overlap between the two, then we can say maybe this was probably random and by chance. However, if there is a significant overlap between the two sets, and we find especially a group of genes that's point to a new pathway or has a specific, where the genes themselves have a pathway connection, then we say, oh, this is interesting. Maybe there's something real happening. So basically, what we're going to do in the next steps is run the same gene correlation analysis for SLU, do our analysis for SLU7, and SLU7 actually the top hit is check1. So that's already interesting because we know check1 is a checkpoint gene. So somehow I think this now is a smoking gun already. So both ATR and check1 correlate with SLU7. That means splicing and the checkpoint must be connected in some way. So this is definitely worth exploring further. Here, this gives you the beauty of having functions. So we had to write all this gunk here, for to get to the result. But finally, we collected all of the functions into this analyzed gene correlation mother function or main function that takes the gene name as an argument, the original data, the numerical columns, our FDR thresholds, and we can tell it we want plots, so we don't want plots. And then this function now will execute all these complicated steps that are written here above in one go. And now if we want to do it again, all we have to do is call that one function. So that's what we're doing here. So now this gives you is a good example of why writing functions makes a difference. So we don't have to copy all this code and change all these parameters. We have just the top parameters to change, do 7, and we run this all again, and it'll give us the significant genes and the top gene, which is check1. So now when we write this, we'll get a lot of printouts here, which gives us the strongest correlation. So ATR is obviously in there because if SLU1 correlates with ATR, ATR is also supposed to correlate with SLU. But there is actually even stronger correlations with check1 and some other genes. So now the question is, we have two sets of genes now, the SLU7 correlations and the ATR correlations. So now we want to look at the network overlap. So what are we doing? How are we doing this? And for this, we're using a technique called Venn diagrams to just display the overlap. And then we'll do a quick statistical test in the end. This Venn diagram overlap is basically you're showing a circle with the size of that is related proportional to the size of the genes you have in one set and in the other set, and you see the intersection here very nicely. The higher the intersection, the more overlap you have. So this is relatively easy, but there's two new things that you need to learn in order to to perform this this analysis to generate these plots. And that's first we need to learn about sets. So this is a very important concept in programming, and this will be applied here. And then we have to learn a new matplotlib based library to generate these Venn diagrams. So let's do this next.